Hello. So in today's class, I'm going to talk very quickly about the biology course, and then I'm going to go through exam terms and what we actually want, and then the paper, how you approach the paper and each section of the paper. So Unit 1. Unit 1 consists of scientific method, food and biomolecules, and everything related to ecology. Unit 2, we have cell structure, then we have our biochemistry, enzymes, photosynthesis, respiration, diffusion and osmosis, cell division, and then all genetics. Unit 3 is the organism. We have our microbiology, we have all our plants, and then all our body systems. So now I'm just going to go through some exam terms and what we want and how to approach these exam terms. So if you're asked to define, write a sentence about that word, what that word means. Give an example if you can, or draw a diagram if you think it will help. So define the cell. It's the basic structural and functional unit of an organism, and it shows the characteristics of life. An example, a blood cell. Function. Say what the job is of whatever that object is. For example, give one function of the kidneys. So the kidneys get rid of waste urea from the body. State. Give that word or that phrase or what they're asking for. State the function of a chloroplast. Photosynthesis. Explain. So tell it in your own words how something happened. Explain how you varied the pH in an enzyme experiment. So you would have used different pH buffers and added that to different graduated cylinders. List or give. So write the words down one under the other. So list or give the products of photosynthesis. Glucose, oxygen. You don't need to say anything else. Outline or write a short note. So write the main points in a list, again using diagrams if you can. If you see outline and or write a note, at least a minimum of 9 marks is going for that. A minimum of 9 marks. So with that, we get 3 correct points. So example, outline the stages in nutrition: ingestion, taking the food into your mouth; digestion, breaking the food down into smaller pieces; absorption, the movement of the digested food in the bloodstream; assimilation, the taking of the digested particles into body cells; and then egestion, getting rid of the undigested food out of the body. Now there was more than, there's 5 points there. So if there was 9 marks, you would have received your 9 marks, but it's a minimum of 9 marks going for outline or write a short note. And answer the 2 things that you've been asked. Example, define excretion and egestion. So 2 definitions here. Excretion, removal of waste products of metabolism. Egestion is removal of undigested food from the organism. Or do one of the things asked. Define excretion or egestion. So you just need to define one. Describe. Give a full detailed account. Use labeled diagrams if you have to, okay, if you think it will help. Describe, again, a minimum of 9 marks can be going for describe. Depending on the sentence, it can go all the way up to 18 marks, possibly even more. So describe physical and chemical digestion in the mouth. So in the mouth, the teeth work on different food in different ways. So therefore you could describe the different teeth in the mouth and their function. The only chemical digestion in the mouth is caused by the enzyme amylase, breaking down starch into maltose. What you could also do there is draw the enzyme amylase, a substrate, the enzyme substrate complex, the enzyme remaining unchanged, and your product, maltose. The physical digestion increases the surface area for the enzyme amylase to work on. So quite a lot of detail in describe. Distinguish between. So write a sentence for each, making sure you point out the differences between the 2 words. A very, very common mistake from students. They'll just talk about one and not the other. So example, distinguish between excretion and egestion. Excretion is the removal of waste products of metabolism. Egestion is the removal of undigested food from the organism. So this year, it was one of the distinguished questions was distinguish between fats and oils at room temperature. So a lot of students said, fats are solid at room temperature, oils are not. So the student did not clearly distinguish what oils are at room temperature. So fats are solid at room temperature, oils are liquid at room temperature. Okay, so you will get maybe a few questions to distinguish between two things. Explain what each one is. Name. So just write the word, you don't need to write anything else. So name one cell organelle that contains DNA, nucleus. Why? Explain the reasoning behind the word or the reasoning behind that process. So why is photosynthesis an anabolic process? It's an anabolic process as it takes smaller molecules and makes them into larger molecules and requires energy for this to be done. Draw a label diagram. So if you're going to draw a label diagram, which you would, I would strongly advise you get a decent pencil. Okay, name the parts, and when you're naming the parts, make sure you bring the line to that part. Don't just, if you're going to draw it, if I just get my pen, don't just draw the line to that and say that's the substrate. Bring your line to what you're pointing at. Okay, so draw a label diagram of an enzyme and its corresponding substrate. For example, this is it here. So don't worry about, okay, if I draw in pencil, my diagram's going to get smudged, it's not going to get scanned properly. It will be. So your paper will be picked up off you on the Tuesday at 5 o'clock, and your paper will be sent down to Athlone and it'll be scanned that Wednesday, Thursday, they'll start scanning the papers. So can you do a pen? Yes, but a pencil. Do you have to be an artist to do this? No, because I'm certainly not. Okay. So remember, always give an example, and if you can, draw a diagram. Now the paper, your exam layout, it's divided into 3 sections, section A, section B, and section C. The overall marks is 400 marks, and it's a 3-hour paper. What you have to look at now is 3 marks is the magic number in biology. 4 marks is going to work out as 1%. How the marking goes, it's not by percent. Okay, so you're going to see your H1, H2, H3, whatever it is in August or your August 2025. You're not going to see an actual percent. So you'll see a number. So the H1 will be from 360 marks to 400. A H2 is 320 to 359. So here's my H1 and my H2. My H3 is 280 to 319. My H4 is 240 to 279. My H5 is 200 to 239. Then my H6 is 160 to 199. Your 7 is from 120 to 159. And anything under that, between 0 and 119 is your H8. So when we're correcting the papers, we don't see a percentage. We just see a number. That's all we see. We see the dotted number at the end. So I could get 360 and I got my H1. Some can get 400 and they've got their H1. So just be aware that is the way it goes. Okay, so it's not based on a percentage. It's where does your number fall within this range to what grade that you're going to get. I would advise my students in your first 5 minutes when you get the paper, open it up obviously, and you're going to read the exam. Then I'd say the next 5 minutes, you're going to read the paper again and you're going to start making selections. And questions will jump out and you'll be like delighted that they're there. So circle those numbers. At the end, if you have time left over the last 5 minutes, which you will read through your answers. So section A is the short questions. So we have 7 questions in section A. So your best 5 are put towards your first 100 marks. So this is worth 30% of your paper. How it goes is, There has to be 2 questions from Unit 1. Has to be 2 questions from Unit 2, 2 questions from Unit 3, and then with this 7th question, it can come from any of those units, but for the last couple of years, it has come from Unit 3. So we know food and biomolecules is going to be from Unit 1. Ecology will be in there from Unit 1, or this year was scientific method. Unit 2 is all your genetics, your respiration, photosynthesis, and then anything from Unit 3. So I would, if you can, strongly advise you to answer all 7 questions, and or 6, but your best 5 are picked. I would leave no blanks. You don't need, no long explanations are needed. We want short, concise answers. Be neat and tidy, and make sure you practice sample paper questions, previous exam paper questions for this. 30 minutes, no more on section A. If you find that you've gone over 30 minutes, stop, leave it, and go on to section B. So section B is all our experiments, okay, 8, 9, and 10. You are asked to answer 2 questions from section B. Some of you will answer 3, that's perfectly fine. So you can answer 2 questions from each one. So this is worth 15% of the paper. And I would advise students, again, no more than 30 minutes on this paper. So you should be nearly an hour into your paper at this stage. What we do know, we know now from question 8 can be from one of these experiments here. Your food tests, ecology, the microscope, and DNA isolation. So DNA was last year. I would say it's highly unlikely that they'd put it back in and repeat it again. But they have to answer, question 8 has to be one of these questions, or it could be a mixture. Now with section B, what you have to be aware of, there's 3 experiment questions. Usually one is just straight on that. So isolation of DNA was a plant was just question 8. Question 9 was just was a bit of a mixture, and then question 10 was just one question. So of that, 8, 9, or 10, I don't know which one. One of those will be a mixture of these. Not all, but maybe some. Question 9. So question 9 is we have our 4 enzyme experiments. We have our photosynthesis. We have our to demonstrate osmosis and prepare and show the reduction of alcohol by yeast. In 2024, it was all about immobilized enzymes. But be aware, there's 4 experiment questions there. With to demonstrate the activity of osmosis, I don't think I've seen it be an actual full 30-mark question, more of a mixture in with questions. Of question 10. Question 10 has to come from one of these. Investigate the role of leaf yeast, you examine the TS with dicot stem, your heart dissection, effective exercise and pulse rate, the effect of IA and growth regulators, germination, and use of starch skim to show digestive activity of during germination. This was 2024. To show digestive activity during germination. Excuse me. Do I think it will appear again? No. But what I do say to my students is, excuse me, just because an experiment question doesn't appear in section B does not mean that it's gone. Some experiment questions can appear also in section C of the paper. So just be aware of that. So if a question asks you to describe an experiment, follow some of these steps. Draw a well-labeled diagram showing the setup of the apparatus. Make sure you describe the control you used. Put the control in the diagram. Describe how the control experiment was set up. Describe the experimental procedure in the correct order. And describe and compare the results. So separately draw your conclusions to the experiment. Always state that you should repeat the entire procedure several times to show that the results are consistently true. So, like I said, answer 2 questions. If you answer 3, perfectly fine. I would also say to my students, actually answer 3 and they'll pick your best 2. Leave no blanks. No long explanations are needed. Short and concise answers. Be neat and tidy. And again, make sure you practice all sample paper questions. Section C. Section C is going to be from question 11 till question 17. So now you have so much more choice since COVID has occurred. So each question is now going to be worth 60 marks. So at the beginning, when I said to you, you're reading through your paper and then you read it through again and some questions will have stood out to you. This is where the questions will have stood out to you. In questions 16 and 17, there's now going to be parts A, B, C, and D. So you have A, won't work, A, B, C, and D. So for example, I'm just throwing things out there. A could be viruses, B could be bacteria, C could be monorail, D could be the digestive system. You only need to do 2. Now, what happens if you like 3 of them? Yes, answer the 3. And your best 2 from this are picked. But then you might go, actually, I can answer all 4 of those. Answer all 4 of those then. And the best 2 will be picked. So that's questions 16 and 17. So they will be 2 30-mark questions. So each question here is worth 30 marks. And your best 2 will be picked for that. Of section C, 1 question has to come from unit 1, 2 questions have to come from unit 2, 3 questions have to come from unit 3, and the 7th question could be from any unit. This section C is worth 65%, and I would strongly advise you to spend 2 hours on this section. If you're answering 4 questions, you're spending no more than 30 minutes on the question. If you find that you've gone over 30 minutes, stop, leave it. Leave a few pages and then go, if you have time at the end, come back to it. So read the questions carefully and look for hints within the question as to how to present your answer, i.e. should you have a labeled diagram, write notes, using a chart, etc. I strongly advise my students to use a black pen. Now think about it, every single book that you read and text, it's all in black because it's easier on the eye. Yes, you can write in a blue pen, but to be really honest with you, if I'm staring at a computer, black, black, black and blue, I actually do have to get away and step up and rest my eyes. A black pen is easier on the examiner's eyes. Another thing that we do like is that for each question you use a new page. So if you answered question 11, answer all question 11. If you go on to a second page, do that and leave it at that. Okay, even if it's 1 or 2 sentences. It's so much easier correcting that. And what you want is, you want the examiner to be happy and go, oh yeah, it's neat and it's clear, it's neat and it's laid out very clearly. So I strongly advise new question equals new page. And if I go back up here to question 16, so if a student answered 16A, I would say leave 16A on the page. Turn the page to 16B. New question, new page. New question, new page. And if any of my students are listening to this, they'll be laughing at that because I do say that. New question, new page. And I try and drill that in from 5th year. Okay, next point here. So make sure you read the question carefully and give the answer that the question demands. Not simply writing down all the information that you know. Answer what you are asked. Don't go on a rant, don't go on a ramble. Use the key words in the question. So throughout your answer, keep referring back to the question so it's keeping you on track, so you're not going on a ramble. So when your answer is complete, check that you've not forgotten to answer any part, that you haven't missed any part. And when students go on a ramble, what it really shows is you don't know what you're being asked. You're hoping, if I put some stuff here, that might be on the marking scheme. So a ramble indicates you don't really know what you're talking about. You're hoping you get a mark. Time management, I've mentioned. So time management is very important, as each long question is worth 60 marks. So I would, like that, assign yourself 30 minutes per long question. And when that time is up, leave that question and go on to the next one. So when leaving on a finished question, leave space so that if you have time at the end, you can go back and complete it. There's less emphasis in the syllables on drawing diagrams, but this is what I believe, they are vital for recording information. and are essential for study and for your revision. So you do not have to be an artist to produce neat diagrams at all, because I am not an artist. And you'll see that some diagrams I've already drawn, not great. Some I will draw in the future, won't be great either. So diagrams should be large and well labelled with the parts in the correct proportion to each. If you're asked to draw a diagram, I would give it at least half a page and put it in the middle, so that you have space either side to label it, or there could be parts to that diagram where you may have to give functions. Use a pencil for neatness and ease of correction. So do not colour the diagram in, as it's a complete waste of time. Don't add shading. It's a complete waste of time. So you're more likely to be asked to identify parts, structures on a given diagram. And how we correct diagrams is, the first thing we have to do is look at the diagram. Does it look like, for example, you're asked to draw the heart. Does it look like the heart? Can you see the ventricles? Can you see the veins and the arteries? So that's the first thing. So there'll be marks going for that. Then it's the labeling. So are the labels in the correct positions? So with diagrams, it's look at the diagram first, ignore the words. Is it what it's meant to be? And then we look at the labels. Graph questions. So you will get a graph question. A graph question will appear somewhere on the biology paper. So either drawing the graphs or the interpretation of the graphs. So should always be answered as marks, or can be easily gained for this. Now I have here, you can get extra graph paper if you run out of paper, but within your booklet, the first few pages is graph paper. Always label your X and Y axis, because there will be marks going for those axes. If it does not indicate that the variable that is controlled along the x-axis and the variable which will change will be on the y-axis. Label both axes clearly, give the units of the measurements asked. Plot if you have to plot this graph. So plot each point accurately with the dot, then join the dots. No ruler, okay? Use with your hand. So plotting the sets, I have here, never use a ruler as it does not follow a biological variation. So if plotting two sets of results on one graph, plot one set of results on the other with an X. Or if there's two lines, do a dash line if you have to. Or use a different coloured pen or a pencil and indicate on the side what each one is. So do not extend the graph beyond those points. So hopefully that has cleared some things you might have been wondering about the paper. But I will go through this again as you approach your mocks and as you're going to approach the Leaving Cert paper. So I'll talk to you again soon. Bye bye.